genetic flow. So how do genes flow through organisms, and then sources of genetic variability. There's all different ways for to acquire variability in genomes, and we're going to talk about some of the ones that are the biggest fields. We'll talk about how new genes are generated, and anytime you generate a new gene, you're usually disrupting a gene that was already there. So those concepts are pretty connected. So generation of new genes and disruption or loss of an existing gene. And then what we will definitely not get to today, but next time we'll talk about the basics of gene expression, going from nuclear RNA to messenger RNA to protein and protein folding, the basics before we get into really the nitty-gritty details in lectures, later lectures. I think 6, 7 is where that starts. Okay, and this image, I always have images that I think are beautiful images of science. I love fluorescent images. And what you're looking at right here is a fluorescent staining of a few cells. So what's in blue? Nuclei, yeah. So there's this dye called DAPI that fluoresces in blue, and it stains nucleotides. So whenever you see this bright blue stain in a cell, you can bet it's probably the nucleus. And then in red, that's actually staining the mitochondria, right? So I like this image because the first thing you might say is, holy crap, cells have a lot of mitochondria. In those diagrams that I see, it shows like 4, right? And so in real cells, cells have a ton of mitochondria, depending on the cell type. In the learning module, it touches on how muscle cells and neurons actually require more ATP than other cells. Mitochondria are the ATP generators of the cell. Those cell types have a lot more mitochondria. Mitochondria often have as short of a lifespan as 7 days. So we're constantly producing new mitochondria. And they actually create more mitochondria via binary fission, right? In the learning module, it talks about the theory of endosymbiosis, how mitochondria, the evolution of mitochondria, there's a lot of evidence that indicates that mitochondria arose from a bacteria that was engulfed by a primitive eukaryotic cell. And so that's why mitochondria have so many similarities with prokaryotic cells, including that they are produced by binary fission. Fun facts. And then in green here we have components of the cytoskeleton, which you will get to know and love much later on in this semester. Okay, here's our review that you are going to diligently read. Or maybe you've already read and studied it. All right, excellent. Okay, so let's start out. Natural genetic flow. I just don't know where to stand in this classroom. I'm so just getting used to it. Okay. I don't like having to stand on one side of the class. It feels, hello, students, like moving around. It feels weird over there. And I know, first world problems. Okay. I say that to my kids all the time when they're complaining about stuff. I'm like, this is really a first world problem. And they don't care, and they continue to cry because their special heart dress isn't clean yet. Okay. So natural genetic flow. When we talk about genetic flow, what that means is how genes move within the population, right? And the two types of, the two categories are either vertical transfer or horizontal transfer. Vertical transfer is very basically parent to progeny. So it is always coupled to cell division or reproduction, always, always. So that can be reproduction of a person to their child, or it can be a bacterial cell dividing, right? Parent cell to daughter cell. That's vertical transfer. So horizontal transfer is between organisms, mature organisms that already exist. Right, so I don't perform horizontal transfer with my children. They're the product of vertical transfer only. So we'll talk about some examples of horizontal transfer in relation to bacteria. So it turns out bacteria use both vertical and horizontal because, right, bacteria divide via binary fission to produce daughter cells. That's vertical transfer. They replicate their whole genome and it is given to the daughter cell. But they also participate in horizontal gene transfer, and there are two types. One is referred to as bacterial conjugation. So some bacteria have what's called an F plasmid, and that is a really small piece of DNA that's in addition to the larger bacterial genome. So say this is my bacterial cell. Here's like the larger bacterial chromosome. Some bacteria also have what's called an F plasmid. So DNA, this is also DNA. And what they can do is replicate that and transfer it to another bacteria that is already exists, right? So this cell's not dividing in this case. What happens is it extends something called a sex pili. Sex pili. And it can reproduce an F plasmid, and the F plasmid can travel through the sex pili, and now this bacteria that has started out just having its own bacterial chromosome will now gain an F plasmid. Gains F plasmid via bacterial conjugation. Like the closest bacteria come to having sex. Okay, so that's one way, right? But the take home really are not details of sex pili and F plasmid. I'm not a microbiologist here. This is kind of the extent of my knowledge about this. The take home really is the fact that this is being horizontally transferred to a bacteria that a totally different bacteria. It did not arise from cell division, right? From this other bacterial cell. Another type of horizontal transfer that happens in bacteria is due to a type of bacterial virus called bacteriophage, the type of virus that specifically infects bacterial cells. And that's what these cool images are over here. So this is a bacteriophage virus. What you're looking at here is a bacterial cell with a bacteriophage virus. It looks like a spaceship landing on it. It inserts its genetic material into the bacterial cell, right? Which is what a lot of viruses do when they infect your cell. They insert their genetic material into the cell, and that reprograms the cell to become a virus-producing machine, right? And so when that cell becomes a virus-producing machine, it starts replicating the bacterial DNA and then packaging the bacterial DNA into new viral capsids, right? These are all viral capsids that are going to be released from that cell. Sometimes when they package the DNA into the viral capsids, they'll take some of the bacterial DNA with it, right? So if they take some of the bacterial DNA with it, and then go infect another bacteria, and insert the viral DNA, they're also going to insert that bacterial DNA that came along for the ride. So that's another way that horizontal transfer can happen in bacteria. It's thought also that in humans, viruses also can horizontally transfer DNA. So have you guys heard of retroviruses before? So yeah, what's one retrovirus? HIV, right? HIV is a retrovirus. So these are viruses that their genetic material is RNA. When they infect cells, they undergo reverse transcription. That RNA is reverse transcribed into DNA, and it gets embedded into the host cell genome, right? So and that is a virus that is infecting a cell that already exists and inserting DNA into that genome. That's the type of horizontal transfer. Does that make sense? Okay, so get the difference between vertical and horizontal. Big thing, if you just keep in mind the idea that horizontal transfer has nothing to do with reproduction or cell division, and vertical transfer has everything to do with it, you're golden. All right. So vertical transfer in sexually reproducing organisms, when we produce offspring, by and large, we are using vertical transfer, right? And the vertical transfer that sexually reproducing organisms use allow for increased potential genetic variability. We as biologists, we love genetic variability. People who don't appreciate genetic variability do not understand biological principles. That's why I feel like if everyone understood science, we would combat racism and we'd be done. Right? Spread biology throughout the world. So there's this really cool story about how we're kind of evolutionarily hardwired to try to attain genetic variability, right? And so the more variety you have in your immune system, there's a complex of genes called the MHC complex. And the more varied the genes are within the MHC complex, the more varied the response of your immune system can be, right? So in theory then, the more variation your progeny has in the MHC complex, the better they'll be able to combat a variety of pathogens in the world, right? Increase survivability. Good, you like surviving. So there's this really cool story or study that was done years and years and years ago. It's called the T-shirt study, where they wanted to figure out the molecular basis of pheromone-based attraction, right? And so what the scientists did is they had a group of men wear white T-shirts, or just T-shirts or whatever. And they told the men, you can't wear deodorant, you can't wear cologne, you can't smoke, you can't do anything to perturb your natural man instinct, right? They just wanted natural man instinct to just fester. And then so what they did is they took those shirts after they'd worn them for an obscene amount of time and put them in boxes and let the stink fester even more. And then they cut holes in the top of the box. And they brought in a whole bunch of women and had them put their faces in the box and rank how attracted they were to the man stink, right? And so then they did these genetic profiles. Dude, you know it don't smell good. Anyways, but they did these genomic profiles of the individuals. And what they found was that women were most attracted to the stink of the man that had the most different MHC complexes from them. Right? So it was this idea that really what we're attracted to is a mate that's going to give us progeny with the best chance at surviving. Of course, we do things that totally get away from pheromone-based attraction because most people do things like wear deodorant and try not to have your natural stink emanate in the world. But anyways, it's kind of an interesting study. So genetic variability is a good thing. And so how does sexual reproduction get at that? So there are kind of three key ways. So first, chromosomes independently assort when they're at the metaphase plate during meiosis, right? So what independent assortment means, or they independently assort as they segregate away from the metaphase plate, I should say. So, right, during meiosis, you have the maternal and paternal copies of each chromosome aligned, right? So, chromosome 1 from your mom, chromosome 1 from your dad, chromosome 2 from mom, chromosome 2 from dad. And the way those get pulled apart in the daughter cells, the way one sorts into the daughter cells, is totally independent of what happens with 2, right? So what I mean by that is I guess I'll be gender normative and do pink and blue, so lame, but probably helps. Okay, this is no longer bacteria. This is a dividing germ cell, right? And so we're just gonna do 2. It's gonna be easy. And we're just drawing them as lines to also make it easy. So we have, here we have our maternal copies. Here we have paternal copies. Chromosome 1 and chromosome 2. Right, so the possibilities here for the daughter germ cells, there's going to be 4. Chromosome 1, chromosome 2, sorry for mixing up the color scheme a little bit on you. Right, so you can have both maternal, you can have both paternal, or you can have one of each, right? So the way those assort, chromosome 2 doesn't care what's happened with chromosome 1, right? So they all assort independently. So if you just had 2 chromosomes, the possible combinations or the possible variation in the sperm or egg cells is 4. We have 23 chromosomes. So the way the math is done there, right? So homologous pairs of chromosomes orient randomly during metaphase 1 of meiosis. Each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently. So the math there is 2 to the n, right? So 2 to the nth power being the number of chromosomes. We have 23 copies. That means for each one of your sperm or egg cells, there are 8.4 million potential ways to combine your chromosomes, okay? So if you already have kids, or if you plan on having kids or not, whatever's your choice, but the egg or sperm cell that you produce, is a 1 in 8.4 million chance of you producing whatever child that you end up producing. That's just on your side, right? So there's a lot of possible ways to combine your chromosomes. So that's one place where you can get variability is independent assortment of chromosomes. Then there's random fertilization, right? So because any sperm can fuse with any egg, that means for each one of my eggs, there's 8.4 million potential combinations. For a sperm, 8.4 million potential combinations, right? So for Emmett and Ellie, right, there was a 1 in 70 trillion chance that I would get my Emmett. There was a 1 in 70 trillion chance that I would get my Ellie, right? And so I always make this joke, but you know how people, there's that like phrase like, oh, you're 1 in a million? If someone says that to you, you can be like, based on the principles of genetic variability, I am technically at least 1 in 70 trillion. Thank you very much. All right. So far we have independent assortment of chromosomes, random fertilization. The other biggie is really hard to quantify because it happens all the time, but we have no way of knowing to the extent that it happens anytime meiosis occurs. And that is crossing over. So crossing over, I hear you learned about in genetics. I wasn't in there with you, but I have it on good, what's the word I'm looking for?
Good faith.
Yeah, good authority, good faith, all of those things that you guys learned about crossing over. So crossing over is when you have that alignment of the maternal and paternal chromosomes at the metaphase plate. They can cross over and exchange genetic material. So what you can end up with are hybrid maternal and paternal hybrid maternal-paternal chromosomes, right? And so what that means is, depending on where that crossover occurs, you might get an allele of a gene that's half long, half deaf, right? So you can, for every gene, there are so many different alleles, right? Look around, we can all see like all these outward differences that we have. There's a lot of inward differences as well. There's a lot of variety out there in the alleles for any given gene, right? So crossing over can create hybrid maternal alleles. So that's another potential source. And so what I want to say in this slide as well, it's showing something called homologous crossover or equal crossover, right? So that means that the chromosomes have exchanged an equal amount of DNA. Sometimes something called unequal crossover happens, and that's going to become really important very quickly in this lecture. So right here you're looking at homologous crossover, and it's when you have an equal exchange of DNA. Okay. So let's start talking about generation of new genes. How do genes change? How do we have all these different alleles within our population, right? So first and foremost, sentence at the top. New genes are made from pre-existing genes. As far as we know, at this point in our evolutionary timeline, there's no mechanism for new synthesis of genes. De novo is a term that you learn in science. There's no de novo synthesis of genes. What does de novo mean? Anew, yeah, from new. Cells aren't from scratch creating nucleotides and throwing them together. What happens is the genome that an individual cell has can be modified in a bunch of different ways. And that's what we're going to talk about. So, where's my remote? Mutation, right? There are different types of mutation. I'll refresh your memory on those different types later on in this lecture. There's various types of errors that alter the nucleotide sequence, right? So I've paired it up so that number one matches the schematic over here. And so yes, sometimes that mutation causes a change that's good. Sometimes it causes a mutation that is detrimental to the organism. Sometimes it's neutral. And a lot of times, you hear the word mutation and people automatically assume like it sounds bad, right? Like, there's all these superhero shows, the mutants are like bad guys, right? I don't know. I'm not good with superhero stuff. Or X-Men, are they good? Oh, okay, good. Okay, that's good. So they're doing good percent. So mutation, whether it's good or bad, I don't know. It's on us to define that, right? There's a lot of genetic changes that make us different. Different doesn't mean bad, right? Like, I think different is beautiful. I think it makes life more interesting. And I think it's really unfortunate that a lot of times what happens when scientists figure out like a genetic change that's associated with a particular phenotype, that all of a sudden people are like, oh, what can we do to fix that? It's like, well, is that something that needs fixing? Is that affecting the lifespan of the individual? Is it causing a person physical pain, right? So, I don't know, that's one place where I think being a good ambassador of science is something that you can do, right? When you hear people talking about mutation, going, oh, that can be a really good thing. That's how we evolved, right? Is by mutations that led to changes in organisms, right? That were, whether it's beneficial or not, sometimes it's in the eye of the beholder. All right. Off my soapbox and onto gene duplication. So gene duplication, right? We're going from one copy of orange gene to two copies of orange gene. Gene duplication is caused by an unequal crossover event, right? In the last slide, I talked about an equal exchange of genetic material. So if you're equally doing equal tradesies from chromosome to chromosome, you're not duplicating anything. If you have a duplication event, that means one of those chromosomes is stealing DNA from the other one and not giving anything back, right? This is a really important driver of genetic change. So we're going to dedicate a lot of today's lecture to gene duplication and how gene duplication can lead to something called gene families if it's coupled with divergence, So divergence, okay, just like Webster dictionary definition, diverge. What does to diverge mean? Yeah, go a different way. So if you have a duplication event, and then these genes, the sequences diverge, they start to become different, which means at least one of them is acquiring mutations that make the sequence different. Now you can start to get, right, new genes that lead to something called a gene family. We'll talk through a bajillion slides dedicated to that. All right. So DNA shuffling is just another term for what I just explained in the last slide. It's equal crossover. So equal crossover and DNA shuffling are synonymous. Yeah. There is, if you take immunology, which so fantastic, really interesting. DNA shuffling is used differently when they talk about how antibodies acquire variation. So that term changes when you get into that specific field, just FYI. But at this level, and with our textbook, DNA shuffling specifically means to, is in regards to equal crossover, which is also referred to as homologous recombination. Recombining chromosomes in a homologous way. All right. Horizontal transfer, right? We talked a lot about that already. So what's an example? Give me one example of horizontal transfer. Bacterial conjugation. Yes, okay. That was just my, are you guys listening? It's lecture 2, so most of you are still listening. It's like midway through the semester. I don't listen to you anymore. Stories of the children. Stop talking about why am I here? So transposable elements. Who here has heard of transposable elements before? Yeah, they're sometimes also referred to as transposons or P elements. These are basically mobile pieces of DNA. The first time I heard about it, it just blew my mind, right? Because I think about DNA within our genome as like it's in its home, on the chromosome, that's where it lives. But transposons are pieces of DNA that can be like, no, I don't like it here, and hop out of that location in the genome and insert themselves somewhere else in the genome on a different chromosome. Yeah, and so it's pretty wild, and so we'll talk about that later too. So those are the 5 biggies, and now I'm going to go into more detail on all of them. And so this is just my reminder to say this, and I already said it, but one more time, keep in mind that many of the same mechanisms that create new genes, they're also disrupting the gene that existed before. So just to kind of have appreciation for that. And so, yeah, which of these mechanisms disrupt existing genes? Yeah, one of the chromosomes just lost that whole gene, right? So that's a loss. Yeah, you've lost gene A and gene B don't exist anymore. You have hybrids of maternal and paternal genes. Horizontal transfer, that's the one where it depends on the type of horizontal transfer. Usually you're just introducing new DNA, but if you're talking like a retrovirus that's inserting DNA and it's inserting right into the middle of an existing gene, that's disruptive. In fact, that's why a lot of retroviruses are the cause of various cancers, because those retroviruses will have there'll be insertional hotspots in our genome, and sometimes if like a retrovirus inserts right into an oncogene, that can be really bad news. Okay. Does anyone have any questions on DNA shuffling? Yeah.
So it said horizontal transfer, they're all, depending on the case, they all have loss.
We're both losing and creating genes, yeah. All right, DNA shuffling, take home, homologous exchange of genetic material has the potential to create hybrid maternal-paternal alleles. All right, let's talk about a super fun one, gene duplication. All right, so, right, you can have these unequal crossover events. And so it's just kind of, you can think of it as just a mistake. Right, in theory, crossing over no, I don't know if you want to think of it as a mistake. I hesitate to use the word mistake. It's a phenomenon in which one of the chromosomes acquires an unequal amount of DNA. So it is not doing fair tradesies, right? It's acquiring enough DNA that you're duplicating regions of that chromosome. And so it can be a small duplication event where you just get multiple copies of a portion of a gene. You can get entire genes duplicated. You can get large parts of a chromosome duplicated. So it just depends. So you have a lot of variety in how much DNA is unequally crossed over. And so what happens there is, we're talking about gene duplication. So the event that you have at least an entire gene duplicated, if not more than one. So if a duplicated gene is necessary, right? Last time we talked about this idea of genes that have critical functions for a cell. They're more likely to be conserved, right? So if we're talking about a gene that has a critical function for the cell, if it's duplicated, why might that duplication now enable development of a new gene? Oh, water break for me, I guess. How about, okay, yeah.
You have two of the same genes. One of them could be made into a protein, and the other one could be, if that one gets mutated, if it's a good mutation, that could be functional in a different way.
That's right. And if it, say the extra copy is mutated in a way that makes it non-functional,
The other gene would still be okay.
you have your backup
It
copy.
would still be able to pass on both genes.
Perfect. Right? So you have a gene that, I mean, let's use a real world analogy. Say you live really far from campus, light rail doesn't make it there, you need a car to get here, right? And you don't want to look like maybe you are fanatical about giants, and you really want to paint your car like bright orange and do giants signs all over it. It's like, oh no, but if I do that, like I don't know what my professor and the classmates are gonna think. So you don't mess with your car, you keep it the color it is, and you keep driving it to campus. But then maybe you get a second car and you're like, I'm gonna do this, and I'm gonna go all in, and you get a fancy San Francisco Giants paint job. And then maybe you find out like, oh, people over here like the Giants too. Then you can think about driving your car, but you have your backup. Where you can, is this not a good example? Should I not have a better example? It's okay. All right. Anyways, the whole idea is you have, if you have a gene, I should probably just go with the biology example. If you have a gene that's required for translation, you need that gene to be functional, right? So it doesn't, if it gets mutated and the organism dies, you're SOL. You're not going to be able to pass on that gene, right? You didn't know what SLL meant? A new type of education for you. You're welcome. That will help you on the exam. Anyways, I shouldn't say, I'm totally going to say that around my kids, and my kids are going to be at preschool being like, SLL. All right. So it's required for translation. You need that gene. If it gets mutated in a way that changes the function and it messes up its ability to perform translation, that cell's not going to be able to survive, right? But if that gene gets duplicated, now you have your backup copy, and this other one, it can acquire changes, and even if it perturbs the way it's performing translation, you have your backup copy that's already performing translation. That was better than the Giants car example. It was funny. I may wish I had a Giants car, I don't know. All right. So having that backup copy allows for more genetic change to occur because it allows for the organism to survive despite any changes in that gene. You have your backup copy. right? And so we see time and time again this happening throughout life. So one great example is hemoglobin, right? So I mentioned last time, we use hemoglobin as an example for so many different phenomenons in this class. And so hemoglobin, right, it's a tetramer. It has two in us, and in many vertebrate or most vertebrate organisms. There are two beta globin subunits and two alpha globin subunits. But if you look way back in our evolutionary history, they're in organisms that are much less complex. They just have a single globin gene that encodes one globin molecule that creates a monomer protein that can bind one single oxygen molecule, right? But throughout time, that globin gene duplicated, and it duplicated again. And so if you duplicate once, you get two. If you duplicate that whole cassette again, now you have 4, right? And so what happened is the beta and alpha subunits are subtly different, and they evolve to have this conformation where they form a tetramer. Each subunit binds oxygen. The way they bind oxygen is pretty fantastic. The first subunit, when it binds oxygen, it binds it pretty rapidly, but it shifts the conformation so that the second subunit binds even faster, the third even faster, and the fourth even faster. And so it provided this really efficient mechanism for hemoglobin to efficiently bind oxygen and transport it through our big old vertebrate bodies, right? So we need to get a lot of oxygen to a lot of different tissues. And so one of the ways that we're able to be large organisms is because of the efficiency that hemoglobin evolved over time through this duplication and divergence of the subunits. Does that make sense? Yeah.
I have a quick question.
Yeah.
So when it comes to gene duplication, that's the idea that the chromosomes cross over unequally, right?
You got it?
So where does this backup gene or backup chromosome come from?
So basically in meiosis, say we had a crossover event where basically chromosome, this is not pink, where's my pink? Right, when this germ cell created the 4 daughter cells, say these are all, there's no tail, so we'll just call these all egg cells. And say there was a crossover event where basically this chromosome 1 stole a bunch of DNA from the non-sister chromatid, and so this chromosome 1 lost a bunch of its DNA. So this egg cell probably is not going to survive. That's the egg cell, right, that a sperm cell fuses with. It's probably going to be, let's stick with my theme, SOL, right? However, if this is the cell that then the sperm fuses with, now you have a scenario where you have a chromosome 1 that has a bunch of extra copies of the alleles that are on that chromosome. It has extra DNA, right? So to be simple, let's say we have gene 1 and gene 2. Here we now only have gene 2, and up here, this one has acquired an extra copy of gene 1. Does that make sense? So this one lost gene 1, this one gained gene 1. Right? So if this was, maybe gene 1 was globin, and now this egg cell has 2 copies of the globin gene. Does that make sense? Okay. Does that make sense, everyone? Okay. Right, again, when we're talking about the acquisition of a new gene and a new function, this is not acquisition of new function that's dependent on mutations that create totally different alleles. That takes evolutionary time. That takes thousands and thousands and millions of years, right? This isn't like a single generation thing. The evolutionary time is really hard to wrap your mind around. It's like thinking about infinity, which hurts my brain. My son keeps asking me about infinity. I'm like, can I just read you a story? Like, it's just hard. Like, can you count to infinity? I'm like, it's a concept, not a number. And he's like, so you can't count to infinity? I'm like, because then he takes his, like, you don't know how to count to infinity, Mom? Lame. But right, like evolutionary time, thinking about what happens to organisms over millions of years, it's hard for us to appreciate. What's our lifespan, right? I mean, it's hard to appreciate that. Okay, so let's talk about how this can lead to gene families. Feel free if you're still confused about that to ask questions. I'll stay on that all lecture up there. So a gene family, the formal definition is a gene family is a group of genes that arose from the gene duplication of a single ancestral gene. If you're a member of a gene family, you're going to have similar DNA sequence and usually really similar biochemical functions, right? So think about alpha globin and beta globin. They bind to oxygen and help carry oxygen. Their sequence is a little bit different. If you think about, I'm going to talk about ABC transporters. I talked about CFTR last time, the gene that's responsible for, when it's mutated, can lead to cystic fibrosis. It's responsible for transporting chloride ions across cells within the lungs. So there are a ton of different ABC transporters. You're an ABC transporter. ABC stands for ATP binding cassette, right? They utilize the ATP, they hydrolyze the ATP in order to transport something across the membrane. But there are ATP transporters that live in the plasma membrane, that live in the endoplasmic membrane, right? That transport all sorts of different things. So to be an ABC transporter, you have to have some level of sequence conservation, right? So back to that concept. You have to be a plasma membrane protein. You have to be something that transports something across a membrane, right? So it's a similar biochemical function. Does that make sense? You're not talking, you don't get to be an ABC transporter if what you're doing is transcribing DNA. It's a totally different function. It's similar functions. And they're going to have similar sequences because they arose from a gene duplication event. In the beginning, they were the same, and then acquired mutations over time. That led to what is it that a mutation causes that leads to a different function? What does it cause to be different in the protein? What equals function in protein?
Structure.
Yeah, structure. So if you start to diverge, you start to acquire mutations, it causes the proteins to have different structures, different functions. So similar but subtly different functions. So I'm going to use a bunch of examples of that to try to just really drive that home. So first, let me orient you to this crazy picture that you're looking at. So up here, this is what a fruit fly embryo looks like. And this is what a mouse embryo looks like. Oh, it's funny. These are two model organisms that the two I've worked with. Much prefer working with the flies than the mice. The mice, there's like this, they're fuzzy and they're kind of cute, but they also bite you. I just did not care for working with them at all. Okay, and what's up with the rainbow colors? So what we're looking at here are these genes that are referred to as the Hox family of genes. In Drosophila, it's called the Homsky complex, but these are all Hox genes. So if it's a different color, it's a different Hox gene family member. And they have specific names, which is what these different letters are. But for the intentions of this class, all you need to know is these are different Hox genes. You can call them pink Hox, yellow Hox, purple Hox, orange Hox, I don't care. Just know that these are different Hox genes. And the colors represent where in that embryo is that specific Hox gene expressed, right? And so what you'll notice is each Hox gene is expressed in its own segment of that embryo, along what's called the anterior posterior axis, right? The head to tail axis. If you ever study development, you learn a lot about anterior posterior patterning, right? How you go from an embryo that just looks like a circle to one that has head structures and wing structures and mid structures, right? How does that happen? All right, so what's going on in the mouse, right? Because here we see 1, 2, 3, 4, 5, 6, 7, 8, 8 Hox genes. Holy crap, there's a lot more Hox genes by the time you get to the mouse. And same thing, there are, the colors correspond to where specific Hox genes are expressed along the anterior posterior axis. So Hox genes are all transcription factors. Transcription factors, right, they help transcribe specific genes. And what these Hox gene family members do is they transcribe genes that are responsible for developing the specific structures that need to develop along the anterior posterior axis. So there are Hox genes that are responsible, if you're in a fruit fly, for creating antennae. There are Hox genes that are responsible for developing the specific legs that emanate from the different body segments, or the wings. So the same thing in the mouse, it's just a mouse is a vertebrate organism, it's more complex. And there's more complexity in the Hox gene family because they've duplicated a bunch more. And so they have, they're still transcription factors, but the genes that they're transcribing and their ability to have variation in the way they transcribe genes has evolved. It becomes more complex. But they have the same function, being transcription factors responsible for expressing genes that are important for patterning the early embryo. Does that make sense? Yeah.
So if two members of the gene family, G A and G B part of the gene family.
Yep.
G diverges too much after so many years, would that still be considered part of the same gene family? If they lose the thing of it diverges so much and loses this
The same function?
Yeah, if it loses function.
Oh, yeah, I mean, that's a good question. If it loses its function,
It diverges so much based on.
so it acquired a mutation that changed its ability, like, to bind DNA or something. Yeah, it's probably not going to be it'd probably regarded as, like, a historical relative of that gene family, but, right, it has to come from the same original gene and have a similar function. If it no longer has that similar function, and we're kind of playing semantics here, but I think there's a pretty strong argument for it not technically belonging to that gene family anymore. Yeah. That's something you can have a super fun nerd war debate on like ResearchGate or something, where it's like the most polite debate ever. I see your point, however. Okay, so because of this, the function of a gene can often be deduced from its sequence if it belongs to a gene family. Right, so when I was a grad student, I was given the potential to work on a project or to have a project where it was a gene that it just had what's called a Celera gene number. So Celera was one of these companies that was at the forefront of sequencing genomes, and so it sequenced the Drosophila genome, and all the genes that hadn't been studied before just got a Celera gene number, so CG3707 or something, right? And so I had some clues as to what the function might be, but the first thing I did was I took the sequence, did a blast search. It didn't have a high degree of conservation with, but enough with this large gene family referred to as lipocalins. Lipocalin, fat, lipo, fat, calin, copper. These are proteins that are responsible for binding to lipid moieties of secreted proteins and helping to solubilize them when they're out in the aqueous extracellular environment. So I went, I wonder if this gene is a lipocalin family member. And 7 years later, I was able to definitively say, yes, this is a lipocalin family member. So you get clues about the function of a gene if it has homology to a gene family, right? If you know, like in my example, it had a specific stretch of nucleotides that was highly homologous to something called the lipocalin signature motif. And so, but then, right, the sequence data can only take you so far, gives you a clue. Then that's why you have scientists who can go at the bench and do years and years of research to follow up on that lead and determine does it have this function. So, okay, that's kind of a sum of what I just said. And you can do kind of cool mad scientist experiments with Hox genes. So when scientists were first trying to figure out, they knew these Hox genes had similar sequences, They have similar functions. What they did was they created a mutation where they express, so this is a normal fly head. So the antenna should come out of these antennal regions. What they did is they expressed the Hox gene that's normally expressed in the segment where legs are produced. It expressed those in the head. And these are legs. These are Drosophila legs coming out of the antennal regions, right? So just expressing that different Hox gene totally reprogrammed that region of the developing Drosophila. All right, so duplication coupled with divergence, okay? So the duplication of a gene and then those genes diverging give rise to related genes. These are very common contributors to evolutionary change. All right, so what are some fun facts about gene families? There are about 5,000 protein coding gene families that have been identified in life on Earth. 250 gene families are designated as ancient. For something to be ancient, you have to find it at least in one bacteria, and at least one archaea, and at least one eukaryotic organism, right? So that means it's ancient because it exists in prokaryotic organisms as well as eukaryotic. It might not necessarily exist in every eukaryotic or every prokaryotic organism. If that makes sense? It just exists across domains. So when people talk about genes being ancient, that's the meaning. And I mentioned last time there are 60 genes that we know are ubiquitous in all genomes that have been analyzed. So, right, here you just have to be in at least one bacteria and one archaea. Here, for a gene to be regarded as ubiquitous, it has to be in every genome that we've analyzed so far. So most of the shared ancient families, same as the genes that are ubiquitous in all genomes, they perform really critical cellular functions like division, transcription, translation, and the biosynthesis of amino acids. Okay. All right, so let's look at the scope of gene, like why am I harping on gene families so much? So it's because of their prevalence. It's in any organism you look at, you're going to identify a ton of genes that reside within gene families. So this is one single species of bacteria called Bacillus subtilis. Its entire genome encodes about 4,000 genes. About half of those reside within gene families, right? So if you look at the pie chart, 53% of the genes, they're their own thing. They don't belong to a gene family. But check it out. 568 of the genes are in families with at least 2 gene members. So that means that those genes were the result of a single duplication and then divergence of it. 273 genes are in families with 3 gene members. So they had at least 2 duplication events. 283 of the genes are in families with 38 to 77 members. A whole crap ton of duplication events. To use technical terminology, crap ton. With education you're getting SOL, crap ton. How did I get this job? So the ABC transporter is the winner. The ABC transporter has 77 members in a single bacteria. An ABC transporter is one of those gene families that you'll see in most cellular life. All right. So expansion of gene families gives rise to function. So the more members that you get, the more specialized that the function that gene family performs can get. Right? So if you just have a single Hox gene, how are you patterning the anterior posterior axis, right? If you have 2 Hox genes, you can get a head and a tail. If you have 3 Hox genes, maybe now you can get head, tail, and some arms in there, right? You can start to get variation in what the gene family is doing. And yeah, one of the big important examples of gene family expansion leading to more specialization and complexity is what we see happen with transcription factors. So BHLH transcription factors, is it written anywhere here? BHLH, or basic helix loop helix transcription factors. It's a transcription factor that's just named after its structural components. And it's a transcription factor family that's expressed in a ton of different organisms. But you see a difference in the number of BHLH transcription factors according to the complexity of the organism. So yeast, right, the single-celled fungi, tomato. I like fungi too. It's like fungi, all right. So there's 7 BHLH transcription factors in yeast. So a little bit more complex here. Worms, right? They're multicellular organisms. You see 41. 84 in my fave, Drosophila, 131 in humans, right? So BHLH transcription factors, they're all transcription factors. They all have a similar structure, but you can start to get subtle differences in the sequences where they're binding to DNA. So you can start to get more variation in what genes they're expressing, which means you can get more variation in the cells in which those transcription factors are expressed in, right? So more variety in the family of transcription factors equals more variation in the genes that they can express. More variation and complexity in that organism. Okay, I see the 410 level of excitement in you guys. I totally understand because that's me in faculty meetings. Like every faculty meeting, like midway through, like, well, and don't, that it stays in this room, not tell that to any other faculty, but like midway through, like, okay. I have the same attention span as my children, actually. All right, adhesion and signaling, that's another big one. Adhesion meaning like cells adhering to one another. If you're a single-celled organism, you don't really need to adhere to other cells, but if you're in us, oh yeah, we need our cells to adhere to one another, otherwise we melt into like a pool of cells on the ground, and that's gross. But right, our cells need to adhere. And cell-cell signaling, our cells communicate with each other a ton. And we're gonna talk about why that's so important later on in the semester, but now take my word for it. So those, take my word for it, that adhesion and signaling are way more important in multicellular animals. There are about 2,000 major transmembrane proteins in the plasma membrane in worms, which are multicellular, which are barely present in yeast, which are single cell. So, so that kind of gets at an expansion in a gene family leads to more complexity and function. All right. At least I'm not changing topics, right? It's still gene families, so that's good. That helps you guys out, right? Gene families. So because of gene duplication, and because of this similarity in their sequences, right? They diverge, but a lot of that structure is going to be the same. So you see a phenomenon referred to as modularity of structure. You guys heard the term like modular housing? Those are houses that have like specific modules which are identical. So proteins, their structures also are often thought of as in modules. And what you'll see in gene family members is you'll see modules of the structure that are totally similar. So what we're looking at right here is elastase and chemotrypsin. These are two digestive enzymes that are produced in our pancreas and function in our gut and help digest our food, right? And we love both of them, we need both of them. And they're both serine proteases. So they have this catalytic site that's marked by this red thing. And this is the site where they bind to their target substrate and cleave it, right? And so if you look at this, it looks pretty similar-ish, right? So what happens is the two serine proteases have almost the exact same binding site. So I'm gonna finish up today with expanding on this specific example of digestive enzymes. So I talked about transcription factors, and now let's talk about 2 serine protease family members. So gene family, again, the definition's at the top. What I did here, remember I drew those nucleotides and said this is kind of what you see when you do a blast search? I actually did a blast search and compared the sequences of elastase and chemotrypsin. This is what you get back. So on the top of every line is elastase, and on the bottom is chemotrypsin. So elastase, chemotrypsin. Everywhere you see a line, that means the nucleotide is absolutely conserved. And it turns out they're about 70% conserved. Pretty similar. And indeed, right, they have pretty similar structure and pretty similar function. So they both belong to the serine protease family, which all kill catalytic sites that include an active serine residue. That's how they got their name. The reactive serine residue cleaves peptide bonds of protein substrate. And it's similar biochemical function. They're both secreted by the pancreas into the small intestine. However, that sequence divergence that has occurred causes them to cleave peptide bonds a little bit differently. So chemotrypsin cleaves bonds next to aromatic residues. Elastase prefers bonds next to small hydrophobic residues. Right? So having those 2 different digestive enzymes allows our gut to process the proteins and foods more efficiently, right? Because of the expansion of that gene here. Yeah.